Hello everyone, welcome back to your lecture series on overview and integration of cellular metabolism. We are in week 6, this is lecture number 28 and we will be dealing with a very important topic of biosynthesis of fatty acid and its regulation. So, we are in lipid metabolism and the concepts that we will be covering today are about the fatty acid synthase multi-enzyme complex and how it helps in de novo synthesis of fatty acid. We will be looking at the citrate transport system and how finally the whole thing is regulated. So, it is very important. So, buckle up and let us move forward. Now, looking at the fatty acid synthesis from a historical perspective, it was discovered by the very famous scientist Fyodor Leinen, who was awarded Nobel Prize for his discoveries in this fatty acid metabolism along with Konrad Bloch in the year 1964. He was a German scientist, right. So, you see de novo synthesis of fatty acid means it is synthesized newly, de novo, inside, okay, inside our body from scratch. So, this all means the de novo synthesis of fatty acid. Now, the primary fatty acid, that is the main fatty acid that we will be looking in our class today, synthesis of palmitic acid. Palmitic acid is a saturated 16 carbon fatty acid, right. So, what are the organs or in which fatty acid synthesis majorly takes place? They are the liver, the fat cells, that is adipose tissue, and lactating mammary gland. Okay. These are the organs in which fatty acid synthesis takes place much more compared to all other organs. So, the mainly more than 90% of the fat synthesis of the body, that is lipid synthesis, takes place in these three organs, right. Now, looking at the enzyme, we all know when we are dealing with, we all know now in metabolism, whenever we are dealing with any pathway, the tools by which the path reactions are catalyzed are enzymes. Now, a very interesting thing about this whole fatty acid synthesis pathway is it is catalyzed by something known as fatty acid synthase multi-enzyme complex. You have heard about this concept in pyruvate dehydrogenase reaction. There was PDH complex, alpha ketoglutarate dehydrogenase complex. This is a bigger version of that. Here the multi-enzyme complex consists of 7 different enzymes. It is a large polypeptide which has got 7 different active site. Not only that, this 7 different active site large polypeptide is acting in pair. So, it is present as a dimer. So, these are the constituent names of the fatty acid synthase multi-enzyme complex, or it is often abbreviated as FAS complex. So, what are the names? Ketoacyl synthase, often abbreviated as KS, malonyl or acetyl transacylase, it is often abbreviated as MAT. In some textbooks you might find they are denoting it separately MT and AT, but this is basically one single subunit that catalyzes two different reactions depending on the case scenario. Next one is dehydratase or DH. Next, enoyl reductase ER. Next, ketoacyl reductase or KR. ACP is acyl carrier protein, and last one is thioesterase, again it is abbreviated as TE. So, why I am saying all these abbreviations? Because from the next slide onwards we will be using these abbreviations, but if you are getting confused you need to roll back to this slide and then look at the names in detail, what are the abbreviations that stand for, right. So, this is all about the structure of the multi-enzyme complex. Now, what is the, this is actually the 3D structure, how in 3 dimension the polypeptide chains are associated with each other in real time, right. So, what is the advantage of having this multi-enzyme complex instead of having 7 different enzymes? Why, what is the utility of all these enzymes being together in a large polypeptide? As you can see, these are the advantages that have been laid down. That is, since fatty acid synthesis is occurring in cytosol and it is a vast area. So, the intermediates can go here and there, but the beauty of the multi-enzyme complex is all the enzymes are together. So, the intermediates can immediately react once it has been acted upon by the previous enzyme. So, this is the reactionary feasibility which is hugely increased due to presence of all the enzymes nearby. Otherwise, if one enzyme is present here, another enzyme is present there, it delays the reaction progress, right? And one more big advantage at genetic level is one gene that codes for all the enzymes. So basically, if we can up or down regulate that enzyme, or I mean gene, all the proteins that will be coded, I mean this single polypeptide, will effectively increase the enzymes in equimolar concentration. So all this actually increases the efficiency of fatty acid synthesis reaction. So the ultimate word is increasing the efficiency of fatty acid synthesis or lipogenesis. That is why nature has chosen all of these enzymes to be clubbed together as a multi-enzyme complex. Now, the process of fatty acid synthesis is basically cyclical. We have discussed that we will be analyzing how a 16 carbon palmitic acid synthesis takes place. The whole 16 carbon is not added together, it is repeated in cycles. So in each cycle the chain is starting from 2 carbon, that is acetyl coenzyme A, and 2 carbons are being added sequentially. So in all 4 carbons are added in each cycle. We will be seeing it very soon. Now, why acetyl coenzyme A? From where did you hear the name acetyl coenzyme A? Very common. We have heard it in carbohydrate metabolism during TCA cycle, and very lately you have heard it in beta oxidation, where we are discussing an opposite path. We are discussing a catabolic pathway where the large fatty acid was broken down into small molecule of 2 carbon acetyl coenzyme A. Well, this fatty acid synthesis is not the simple reversal, but the starting material is the same, that is acetyl coenzyme A. So acetyl coenzyme A is the starting material and it is converted to malonyl coenzyme A by the enzyme acetyl coenzyme A carboxylase, and this is the rate limiting regulating key reaction in the whole fatty acid synthesis. Now you might ask or you might wonder, this name acetyl coenzyme A carboxylase is not in this abbreviation, and you are very right, because acetyl coenzyme A carboxylase or acetyl CoA carboxylase is a separate entity. It is a separate enzyme which is not a part of fatty acid synthase complex. Okay. So first, the action of acetyl CoA carboxylase converts acetyl coenzyme A to malonyl coenzyme A. Acetyl coenzyme A is a 2 carbon compound. It uses 1 carbon is added from bicarbonate and it results in formation of a 3 carbon malonyl CoA. But this is the main key reaction, and the cofactor of this acetyl coenzyme A carboxylase enzyme is biotin or vitamin H. Now this acetyl coenzyme A carboxylase in itself is a bit complex enzyme. So let us look how the acetyl CoA carboxylase enzyme acts. Okay. So it's very interesting, you just follow along. First we will discuss about the cofactor biotin plays a very important role. It in between the reaction it becomes carboxybiotin. So at first the biotin moiety is carboxylated and thereafter the carbon molecule shifts to acetyl CoA to form malonyl CoA. So just look at here, the first step of this reaction is basically transfer of the carboxyl group to biotin. So this whole acetyl CoA carboxylase enzyme functions as 3 separate enzymes in itself. The first function is biotin carboxylase. Okay. So the first, with the help of ATP, this is energy dependent process, this HCO3 minus, the carbon from HCO3 minus is transferred to biotin, all right, and hence biotin becomes carboxylated. So this is known as carboxybiotin, right. Now this carboxybiotin undergoes an internal conformational change where the carboxyl group is just flip flopped from the biotin carboxylase domain to the trans carboxylase domain. So basically transfer of the activated CO2, this is the activated carboxyl group from biotin carboxylase region to trans carboxylase active site. Okay. As you all know from regulation of enzyme activity, a substrate always mimics the enzyme's active site. So this acetyl CoA carboxylase enzyme has got 2 active sites in itself which can bind to the carboxyl group of biotin internally. So a transformation occurs and this is the trans carboxylase activity. And in the final phase, then only with the help of this trans carboxylase activity, the carboxyl group is transferred to acetyl coenzyme A. So, a 2 carbon acetyl coenzyme A becomes a 3 carbon malonyl CoA, and the biotin, that is the coenzyme, is again regenerated. So, in the whole thing, basically what happens, acetyl coenzyme A along with biotin, a carbon molecule from bicarbonate comes to from carboxybiotin, and in the next step the carboxyl group goes to acetyl CoA and biotin is regenerated to form malonyl coenzyme A. So, now we have got malonyl coenzyme A, which is actually the form by which the acetyl coenzyme A molecule enters into the fatty acid synthase complex, right. Now, the very starting molecule is of course acetyl coenzyme A. So, in the first step, one acetyl coenzyme A will bind to fatty acid synthase complex, and in the next step a malonyl CoA will bind to fatty acid synthase complex. So, it is all about binding. Now, where does it bind? Those acetyl coenzyme A and malonyl coenzyme A have got affinity towards the sulfhydryl group. Where this sulfhydryl or SH group is located? It is located at the enzyme ketoacyl synthase or KS, and who helps in transfer? That is the enzyme MAT, acyl transacetylase or malonyl transacetylase, and with the help of acyl carrier protein. So, now just see what happens. This is the basic concept where acetyl CoA and malonyl CoA first attaches to the SH group of the condensing enzyme. So, the first step, in the first step what happens? Acetyl group from acetyl CoA is transferred to the SH group by the enzyme malonyl or acetyl transacetylase. I told you the same enzyme codes for transfer of acyl group from acetyl CoA as well as from malonyl CoA, acetyl group I mean. Anyway, so it is transferred to the enzyme ketoacyl synthase. You see over here what is happening. One molecule of acetyl coenzyme A is coming and the coenzyme A part is lost. The acetyl group is being attached to the ketoacyl synthase domain Ks with the help of acyl carrier protein or ACP. Okay. ACP actually helps in everything, you will see. So, this is the first step where the coenzyme A part is lost and the acetyl group CH3CO is transferred to the ketoacyl synthase Ks. So, if this step is clear, in the next few steps it will be very easy for you to understand. So, please make sure you are understanding this whole thing. Acetyl group comes, okay, ACP has got a sulfhydryl group, it helps in attachment. So, ACP actually bends over, it helps in attachment of the acetyl coenzyme A acetyl group to the ketoacyl synthase domain. In the next step, similar reaction happens. What happens? A malonyl group is also transferred to the ACP SH group by malonyl transacetylase. So, the acetyl group was transferred to ketoacyl synthase and the malonyl group is transferred to the ACP. Fine. So, both has got disulfide linkages, I mean sulfhydryl groups. Acetyl group is attached to Ks and malonyl group is attached to ACP. Now, these two reacts very closely. Okay. So, the ACP is actually lying. So, the malonyl group on the acyl carrier protein is lying very closely, is located very closely in 3-dimensional structure to the acetyl group of ketoacyl synthase. Fine. Now the enzyme is actually primed for all the reactions. So, in order for the enzyme to function, it needs to receive the initial group. First, it started with an acetyl group that was allotted to ketoacyl synthase domain and one malonyl group that was allotted to the acyl carrier protein. Now, these two react, these two condense together with the help of the enzyme ketoacyl synthase. Okay. So, what happens? You see, the acetyl group from the ketoacyl synthase is actually transferred to the malonyl group. Now, a bonding has been formed and it has led to the formation of aceto-acetyl ACP and one carbon dioxide is lost. Fine. So, aceto-acetyl ACP, where malonyl CoA was 3 carbon, acetyl CoA was 2 carbon, however, one carbon dioxide is lost. So, now it has become a 4 carbon aceto-acetyl group and it is always attached to acyl carrier protein or ACP. So, aceto-acetyl group is attached to acyl carrier protein. Mind it, when you are learning beta oxidation, we learned everything was in coenzyme A form. In this case, fatty acid synthesis, all the intermediates are in ACP form. It is bound to this ACP, right. So, in the next step, what happens? You see aceto-acetyl ACP, now a reduction happens, reduction with the help of NADPH, very important. So, when an aldehyde or keto group is reduced, it leads to the formation of an OH group or alcohol. So, this is beta hydroxy butyryl ACP. Okay. So, this keto group has been reduced to alcohol. Who has donated the hydrogen? Who is the reducing equivalent here? NADPH, very important. It is not NADH. We discussed what are the roles of NADPH. It was helping in fatty acid synthesis during our HMP shunt lectures. So, now you see this is the step where NADPH is exerting its role as a reducing equivalent, as a hydrogen donor. There are more. So, in the next step what happens? There is dehydratase enzyme action. So, a water molecule simply goes out. When a water molecule goes out, if you can, if you just remove an HOH from here, what will happen? There will be a introduction of a unsaturation or a double bond. This is exactly what happens here, and the resultant compound is known as delta 2 trans-enoyl or delta 2 butanol ACP, delta 2 butynoyl acyl carrier protein, and the enzyme that is acting is beta hydroxy acyl dehydratase or simply DH, right. So, now when the unsaturation has been reached, the next part is again a reduction using NADPH. The name of the enzyme is enoyl reductase. What will happen? The unsaturation will be gone. So, the whole purpose was to get rid of this OH group, you see. So, in order to get rid of this OH group, we introduced an unsaturation with the help of dehydratase enzyme, and again we have reduced with the help of NADPH, and this is now butyryl ACP or a 4 carbon acyl carrier protein. So, a 2 carbon has now been converted to a 4 carbon which is attached to the acyl carrier protein. So, now what will happen? You remember initially acetyl coenzyme A was bounded to acyl carrier protein and it was translocated to ketoacyl synthase so that it could receive another molecule of malonyl CoA. Well, the same thing is going to happen over here. So, now translocation of butyryl group to the ketoacyl synthase domain happens, and finally the acyl carrier group will become free to receive another molecule of malonyl coenzyme A, and a second round of this whole thing will be repeated. So, first acetyl group of acetyl coenzyme A got attached to Ks, then malonyl, a 3 carbon, was attached to ACP, and ultimately those two reacted, and they after a series of 4 reactions they formed a 4 carbon. This 4 carbon is again transferred to Ks, and now again when another malonyl CoA will be attached to ACP, the same series of reaction will happen. One molecule of carbon dioxide will be lost, and ultimately the chain will increase by another 2 carbons. So, this process is repeated for 7 cycles. Why 7 cycles? It started with 2 acetyl coenzyme 2 carbon, 7 more cycle, 14 additional carbon, and ultimately we get the 16 carbon palmitic acid. So, when the desired length is achieved, then only the 16 carbon fatty acid will be detached from acyl carrier protein by the enzyme TE or thioesterase. Mind it, during all this internal cycle the action of TE is not required. TE is only required when the desired length of fatty acid has been reached, and thus we get the 16 carbon palmitic acid with all unsaturated CH3 at the end, CH2, CH2, CH2 in between, and COO at the end, or COOH, right. This is the palmitate icon, palmitic acid is COOH. So, if we look at the stoichiometry or the calculation, so just as I told you, it starts with 1 acetyl coenzyme A, and finally 7 malonyl coenzyme A is added to it, but ultimately, as we saw, in each step 1 carbon dioxide is lost, right. And where from this 7 malonyl coenzyme A came? This was actually by the first reaction where carbon dioxide, this is also known as CO2 fixation reaction, where carbon dioxide in the form of bicarbonate actually converts acetyl CoA to malonyl coenzyme A. So, if we look at the entire thing, this looks like 8 molecules of acetyl coenzyme A along with 7 molecule of ATP, 14 molecule of NADPH, because NADPH is required in 2 steps. So, 7 cycles, 2 steps, 14 NADPH, it gives rise to 1 palmitate or palmitic acid, 14 molecule of NADP plus, that is the oxidized form of NADP, and ultimately 8 acetyl CoA molecule, and you know the others. If you just look at the cycle, you will easily get the stoichiometry, what are the initial and the final compounds. So, ATP hydrolysis happens. This is an energy dependent process. Now, this whole thing, fatty acid synthesis, is exclusively happening in the cytosol. I told you why in the cytosol, mainly because the presence of high amount of reducing equivalent. What is the reducing equivalent? NADPH. So, in cytosol the NADPH is much more compared to NADP plus, but I told you this NADPH is not present in all the cells and all the organs. It is only present in those organs where this fatty acid synthesis is happening more and more, it is happening in excess. So, what are the 3 organs? I told you, in hepatocyte, lactic mammary glands, adipose tissue, these are the organs where there is excess production of NADPH by HMP shunt. These are the cells where maximum amount of HMP shunt is happening, these are the cells where maximum amount of fatty acid synthesis is happening. And I also told you that apart from production of NADPH by the enzyme glucose 6 phosphate dehydrogenase, this is the main source of NADPH production, malic enzyme also plays an important role, right. Now, one question you should be having in your mind right now, that we need acetyl coenzyme A for fatty acid synthesis, but this acetyl coenzyme A is produced from pyruvate, and this pyruvate dehydrogenase reaction happens in the mitochondria, right. So, where from we are getting acetyl coenzyme, or how acetyl coenzyme is coming out from the mitochondria to the cytosol? It is actually not directly coming out because the mitochondrial membrane is not permeable to acetyl coenzyme A. So, it actually gets transferred in the form of citrate across from the mitochondria to the cytosol. Okay, so, this is the basic cycle. We can look at it in an elaborate fashion. This is the same thing that we have elaborated in this figure. So, you see inside the mitochondria acetyl coenzyme A is formed from pyruvate. It now condenses, it is converted to citrate with the help of citrate synthase enzyme. Citrate synthase enzyme is a part of, that is for you to answer, right. So, in the form of citrate, it can come outside into the cytosol. There it is acted upon by the enzyme citrate lyase, very important, citrate lyase. Okay. Here one now with the help of this enzyme, the acetyl coenzyme A is generated back, and it is also an ATP dependent process, and this acetyl coenzyme A is used for fatty acid synthesis, and the citrate gets converted to oxaloacetate. Now, in the next step, oxaloacetate is acted upon by the enzyme malate dehydrogenase, is cytosolic malate dehydrogenase. There is also another mitochondrial malate dehydrogenase, that also you already know by now. So, malate dehydrogenase now is acted upon by malic enzyme to form pyruvate, all right. This malate or pyruvate are actually permeable to the mitochondrial wall or inner membrane. Preferably pyruvate is much more permeable, so pyruvate again goes inside, it gets transformed to oxaloacetate and the whole cycle is completed. Whereas malate can also be transferred in a small amount, and also malate will be acted upon by oxaloacetate to form by malate dehydrogenase to form oxaloacetate, and you know what the cycle is going on over here. So, you can see this all thing looks like a spiral. Due to this layout, the fatty acid synthase complex is also the whole thing, the citrate transport along with fatty acid synthase complex, which was discovered by Fyodor Leinen, is known as Leinen's spiral. Now, as I told you, the fatty acid synthesis reaction is not a simple reversal of fatty acid oxidation. So, there are differences. The very first difference that you are already guessed by now, that is the location. Beta oxidation takes place in mitochondria, fatty acid synthesis takes place in cytoplasm, right. The intermediates are present as coenzyme A, whereas the intermediates in fatty acid synthesis are covalently linked to the sulfidyl group of acyl carrier proteins. All the enzymes of beta oxidation are isolated, they are independent enzymes, whereas it is present as a multi-enzyme complex. 2 carbon units are added in case of beta oxidation, or 2 carbon units are broken down at a time in case of beta oxidation, whereas 2 carbon units are added, but as 3 carbon malonyl CoA, right. Next, the coenzymes over here are NAD and FAD, whereas you already saw it was the reducing equivalent is NADPH, right. The transporter, of course, carnitine pathway is very important to form acyl carnitine. This is the pathway by which fatty acids get inside the mitochondria, whereas acetyl coenzyme comes out of mitochondria as with the help of citrate transporter. And in the regulation, of course, you know by now this is anabolic, so it will be helped by insulin and it will be suppressed by glucagon, and the counter, this is a catabolic pathway, beta oxidation, so insulin inhibits it and glucagon promotes it, right. Now, let us look at the regulation of fatty acid synthesis. As I told you, the main enzyme is acetyl coenzyme A carboxylase. So, if we control the activity of acetyl CoA carboxylase, we can control the activity of fatty acid synthesis. So, how it is controlled? First, end product. So, palmitoyl CoA, when palmitate is formed in excess, we do not need the formation of more palmitate. So, palmitate actually inhibits the acetyl CoA carboxylase enzyme. We all know by the covalent modification, glucagon, epinephrine, that is all the catabolic hormone will prevent this anabolic process, right. But there is one allosteric stimulator. Who is the allosteric stimulator? That is citrate. Okay. If citrate is present in excess, it will help its own breakdown by the help of the enzyme citrate lyase, and more acetyl coenzyme will be formed, and more acetyl coenzyme will now be utilized by the acetyl coenzyme carboxylase, and insulin acts as a trigger for activation. As we all know, insulin being an anabolic hormone will help in the fatty acid synthesis. Not only that, it has been found that the dephosphorylated form of the enzyme is active. Of course, if insulin is activating, it is dephosphorylated, and in dephosphorylated form the enzyme undergoes it polymerizes to form a long filament, and this is the electron microscopic structure of the acetyl coenzyme carboxylase enzyme. So, looking at the regulation in term of short term and long term regulation, the amount of acetyl coenzyme A and the amount of ATP, if it is more, if it is in excess, so what it will do? It will inhibit isocitrate dehydrogenase. Isocitrate dehydrogenase is an enzyme of TCA cycle. It means the body has got more energy, we do not need any more energy, we do not need any more ATP, we need to now store things. So, lipogenesis will be favored, and this actually increases the amount of citrate. Since the enzyme isocitrate dehydrogenase was utilizing citrate, if it is inhibited, citrate will, the concentration of citrate will rise, and this will help in upregulation of acetyl CoA carboxylase, as we saw in the previous slide. And also this citrate is acting as a carrier of the acetyl coenzyme A that we just discussed, right. So, more and more citrate means more and more fatty acid synthesis. We also discussed about covalent modification. Basically, glucagon we all know it how it acts by the cascade. Increased glucagon means increased amount of cyclic AMP. It will activate protein kinase and it will help in phosphorylation of acetyl-CoA carboxylase, and phosphorylated form is inactive. It will be switched off. So, more glucagon means we do not need more fatty acid. Now, glucagon means catabolic fatty acid will be broken down in the form of beta oxidation. I told you palmitoyl-CoA is inhibiting. How it is inhibiting? It, number one, it inhibits acetyl coenzyme carboxylase. It also inhibits translocation of citrate from mitochondria to cytosol, right? Also, it inhibits G6PD. So, G6PD if inhibited, there will be no NADPH, again there will be no reducing equivalent, the fatty acid synthesis will be hampered. And lastly, this is the long term regulation where diet actually plays a very important role, and this is very, I mean, you can guess it from your common sense, since the body has got if the diet is of low fat and high carbohydrate, the body needs more fat, then the activity of acetyl coenzyme carboxylase and fatty acid synthesis will be increased. So, there will be more and more lipogenesis, and if we are fasting and having a high fat diet, then the body will try to break down all the fatty acid, then the activity of acetyl coenzyme carboxylase will be decreased. And what actually happens in high fat low carbohydrate diet, we all saw that it leads to, especially Atkins diet, it leads to production of more and more ketone bodies, because oxaloacetate has been shunted towards gluconeogenesis, there is no TCA cycle, acetyl coenzyme cannot be broken down even if beta oxidation is happening, and ultimately ketogenesis is formed. So, to conclude, this lecture has covered the concept of the fatty acid synthase multi-enzyme complex and how it helps in all the reaction of de novo synthase, what are the citrate transport system, how acetyl coenzyme is getting out from mitochondria to cytosol, and how the whole fatty acid synthesis is regulated. So, these are my references, and thank you for your patient hearing.